Well, welcome to biology class. This is your first lecture with me, and I'm excited to have you on board. We are going to start with the very basics. So let's begin with biology itself. What exactly is it? As most of you already know, it's the study of life, study of living things in a scientific way. So what exactly does that mean? Well, in order to study things in a scientific manner, we needed a method, and of course that is going to be the scientific method. So whether we're studying whole organisms or we're studying specific parts of living things, even down to the chemistry of living things, we'll talk more about that later, we need this specific method to go by. And so just like anything else, it's going to start with observations. Living things have to follow natural laws of physics and chemistry, for instance. And so we first make observations, and then we develop a hypothesis, we test it, and then gather our data. And then we either accept or reject that hypothesis, and then we present that information to the rest of the scientific community for them to scrutinize it to make sure that we have done our proper diligence. So this is just another way to look at that process. So when we are starting to formulate a question in our mind, we are going to do some research first. So typically speaking, if a scientific person comes to any question, they're going to go looking through the vast amount of information that's already been presented in various scientific journals and things like that, and gather the knowledge so that you can start having an informed hypothesis. And we'll talk a little bit more about what a hypothesis is. And then, of course, we'll test our hypothesis using a well-designed experiment. And from that, we get results, and from those results, we draw conclusions. Now, those conclusions can then mean that our hypothesis was supported, or that it was not supported. And if it's not supported, then we can come back up here and maybe come up with another reason as to why it wasn't supported and come up with a new hypothesis. Sometimes that requires us to go back and do a little more research before we can create a second hypothesis. And then we go through this process all over again until we get reliable and repeatable results. Okay, so like I said, we're going to start with observations, and we're going to use all of our senses to do these observations, but then we also can further those observations using equipment, such as a microscope or a telescope, something like that. But here are just some easy examples of observations. So if we observe that the grass on the school's front lawn is wet, we naturally start to make inferences that we can then start working into a hypothesis. So for instance, we could think it rained, the sprinkler might have come on, the dew might be the reason for the grass being wet, maybe a dog peed on the grass. All those inferences that we come up with can explain why the grass is wet. Those inferences are based on prior experiences. And as scientists, we also have those inferences based on the knowledge that we've gained through our research. So all of the reading that we do of past experiments, of natural laws, information that we already know about physics and chemistry, all those come into play when we have our inferences, which are going to help inform our hypothesis. Okay, so now we come to our forming of our hypothesis. And that hypothesis is going to be a possible explanation for the observations that we've been making. And like I said, these are informed based on those inferences. So things that we need to always have in our hypothesis, it needs to be clear and understandable, and it needs to be testable and measurable. It doesn't do us very much good to have some vague answers. That's just not the way the scientific community works. So it has to be testable and it has to be measurable. And it has to contain an independent and a dependent variable. We will talk a little bit more about that. So often we write it as an if-then statement. If such and such happens, then we expect such and such to occur. Okay, that's how we write our hypotheses. Now there are two types of hypotheses that we're going to be using, and the first is what we call the null hypothesis. The null hypothesis. So what we mean by that is we're going to say that if we change something, there's going to be no impact. So there's going to be no relationship between our variables, basically. So in these examples here, our cat shows no preference for food based on shape, or the plant growth is not affected by light color, or age has no effect on musical ability. That's called our null hypothesis, and it's often represented as an H and then with a subscript 0 underneath it. And that just basically says there's no relationship between our variables. Now, our second hypothesis is really what we think is going to happen. This is where our inferences come in, and this is really what we're going to test. So the alternative hypothesis predicts that there is a statistically significant relationship between the variables, or in other words, one variable is dependent on the other. So in our example here, we're looking at plant growth, and we're trying to determine the effects of a biofertilizer on that plant growth. So our two hypotheses here, the null hypothesis is basically going to say this biofertilizer has no significant impact on the growth of the plant. Whereas our alternative hypothesis, which if you'll note we represent usually as H1 or HA, either one, predicts that there is going to be significant growth when we apply the biofertilizer. Okay, so that's specific because it's relating to very specific variables, application of biofertilizer and growth of a plant. And we're stating that there is going to be an effect on the growth of the plant. Okay, so very specific, very measurable. Okay, so now we need to design an experiment to test our hypotheses. So here are a couple of things that we need to do. We need to be able to identify our variables, and then we need to have control groups and experimental groups. So there's the independent variable. That's the variable that I choose to change because I'm going to test it. It's the if part of your hypothesis. So if we were, again, looking at the biofertilizer and the plant growth, we would say that the variable in that is the application of the biofertilizer. So that's the if part. If I apply biofertilizer, then we come to the dependent variable. That's the variable that I measure, or the response. So in our sample experiment, then we're going to measure the amount of growth seen in the plant. So that's the then part of our hypothesis. So if I apply a biofertilizer, then I see significant plant growth. Okay, so those are our independent and dependent variables. Now we need to talk about our controls. You hear that a lot, I'm sure. Well, did these experiments have the proper controls? That means that if I keep the variables the same, in other words, let's say for instance I am in the same experiment, that fertilizer has to be dissolved in water, right? So my control group would then be the part where I just water the plant. So that water doesn't contain the independent variable, it doesn't contain the fertilizer, but I'm still going to measure the dependent variable, which would be our plant growth. So our control group would be just watered with water, and our test group, which it doesn't really say on here, but that's the other group. That test group would be the one that has biofertilizer in it. Now, we can actually add more test groups to it. We could test different concentrations of that biofertilizer and allow our water to still be our control group. So we can have multiple test groups, but we just need to make sure that our controls make sense. For instance, the other thing we need to think about when we're designing experiments is everything else the same? So in this same experiment, if we are growing these plants, the test groups as well as the control groups have to be at the same temperature, the same humidity, the same amount of light exposure. So all of those are controlled variables. They need to be kept the same. So those are important when we are designing an experiment, to make sure that we are testing for the proper thing and that extra variables are accounted for. Okay, so then once we've designed the experiment, then we implement it, and then we start collecting our data and analyzing it. And this can be very fun. It also can take quite a long time because you do have to mathematically analyze things. So a couple of things that you'll need to know when you're looking at your data is that what we want to check for is what we call the p-value. So the p-value in our data is just the probability that the results happened by chance. So if in our experiment we got plants that grew 2 feet because of the biofertilizer and those in the control group only grew 2 inches, well, is that difference because of the biofertilizer, or did it just happen randomly? And the lower the p-value, the higher the confidence we can have in the results. And typically speaking, the more controls you have, the lower the p-value is going to be. So for most scientists, a p-value of less than 0.05 is considered acceptable. Okay, so if we know we have a low p-value and we can have confidence in our data, then we need to look at what the data is actually telling us. So is there a significant difference between the test group and the control group? If there is not, then we're going to accept the null hypothesis, also rejecting the alternative hypothesis. And what that means is that we're going to go back up again in our process and try to come up with a different hypothesis, because apparently that was not, our alternative hypothesis really wasn't accurate. If, on the other hand, our answer is yes, there is a significant difference between the test group and the control group, then we can pretty positively say, for our example, that the biofertilizer had a significant impact on plant growth. Great. So what do we do once we have that information? Okay, we have all of our data. We've decided that our alternative hypothesis was correct. Yes, our biofertilizer significantly impacts the growth of plants. Great. Now we have to present that to the scientific community. How are we going to report those results? First of all, we're going to take all of our data and put it in easily understood forms. So whether that be in bar charts or pie charts or scatter graphs or some table, something like that, we're going to be able to put that together into an article where, along with the words that describe our procedure, how we did it, what the data actually was. In those papers, we usually give a little background as to how we came up with the hypothesis, what the general information is already, like a short summary of the scientific information already available, and then how our data fits into that and the conclusions that we can draw from it. So 2 things about this. One, anytime you are presenting a table or a graph, it should be able to stand alone. That's extremely important to me, and you'll see that when we have labs. I expect titles that make sense. I want the reader to be able to look at the table by itself and know what happened in your experiment. I want those readers to know the results, the conclusions that you came to, and exactly what's presented on your graph. I have unfortunately had some students that would turn in a graph to me that said, for instance, line A is greater than line B. Okay, well, if I looked at that and I had no context as to your experiment, I don't know what line A is, or line B for that matter. So you have to be good communicators in your tables and graphs. Okay, so that's number one. Number two, just for your information, really, every scientific journal that gets published, when the author submits their data in the form of a journal article, it goes out for peer review. That means that people who work in the same scientific field, so they're familiar with the body of work and they understand the concepts, will read over it and can ask questions, can say, no, I don't think that's a good enough control. Have you considered this? And really basically put it through the ringer so that by the time it gets published in scientific journals, it should be something that is reliable and that you can use. So that would be the end point of our whole experiment. So as biologists, this is the process we go through when we delve in to study something about either a living organism, a specific part of a living organism, or even how organisms interact together. So this process, the scientific method, is what is used each time. Okay, so now that you know how we study life, let's just talk about some basic characteristics of life. The very first is that the basic building blocks of all life is cells. So the smallest unit of life is a cell, and we're going to talk significantly as we move through the semester about each part of the cell, how it works, how they work together, and I hope that you are as blown away as I am by the complexity of such tiny little things and how they all work together, and it's really just fascinating and it'll draw you in a heartbeat. So, but that's the great thing about life, is it all starts at these tiny little building blocks called cells. Okay, so the smallest form of life is a cell. Those cells, of course, are made up of atoms and molecules, and then we go up from there. This is sort of the way we classify, organize life in general. It helps us to put things in perspective and helps us to analyze things when we can organize them like this. So at the very bottom, of course, this is where our atoms and molecules come together, and then we build cells. And like I said, we're going to talk significantly about types of cells and how they work together. And then cells come in together to make tissues. So for instance, your skin is a tissue and it's made up of a whole bunch of epidermal cells. And those tissues can then work together to make organs. On this example, like your brain or your heart or your lungs, keeping in mind that plants are also living things, so leaves are an organ on plants. And then that going into your organ systems, for instance, your cardiovascular system. And then good to know something similar to that is a vascular system in plants. And then organ systems building together to make organisms, and then populations, communities, ecosystems, and biospheres. So in 111, we are going to get up to probably more like possibly the organ level, maybe only even the tissue level. If you'll hang with me for 112, we get to do lots of fun stuff at the organs, organisms, and population, community, ecosystems, ecology, and biosphere as well, and that's a lot of fun. So I hope that you will consider doing that with me next semester. But for now, we are starting with the very basics of biology. So we've talked about how we study life, that they are at the very basics cell, but when we talk about we study living things, what does that really mean? Like, if I were to say, is this thing living or not, it has some conditions that have to be met in order to be considered living. So I'm going to list those real quickly, and then we're going to kind of go into it after that. So to start with, all life requires an outside source of nutrients and energy. That's pretty self-explanatory. 2, homeostasis has to be maintained by living things. We'll talk about what that means. 3, living things have to be able to respond to stimuli. You know, somebody pokes you, you move away. That's one example. 4, living things must be able to reproduce and to develop. And then 5th and final, living things must be able to adapt. Now, here's a caveat that I need to throw in here for living things. In the terms that we are using, let's take this tree, for example. I know that this tree meets all of these requirements. It does require an outside source of nutrients and energy. It is allowed to photosynthesize using the external source of solar energy and carbon dioxide, so it meets that one. Homeostasis is maintained. I know you don't know what that means exactly yet, but it knows how to balance its salts and stuff like that. It can respond to stimuli. If you've ever seen root growth, it goes down in response to gravity, and stems grow up in response to light, so it can definitely respond. It is able to reproduce and develop. Yep. And then it's able to adapt. Yes. Okay. But here's the question. If I gave you a piece of wood, would you consider that something that is living or not living? That's a fun question. So I will just go ahead and give you the answer on that one. It is considered living because when we are talking about living things, we mean something that is either currently living or has lived in the past. So when we're looking at fossils, we're talking about that is a living thing, as is this dead piece of wood. That word dead meant that it was living in the past. Okay? Okay, we said that our first requirement to be a living thing is that you require an outside source of nutrients and energy. So nutrients being the chemicals that we need to create, like proteins and your cytoskeleton, things like that. Those nutrients are generally going to be ones that are created at our producer level. So if you're looking here at this diagram, you'll notice that here are our producers. Those are what we usually call them our primary producers. So those are the ones that can use solar energy via photosynthesis to create sugars, which is used as energy, and that energy is used to then create all of the building blocks that we need within the cells. So like I said, proteins, DNA, that sort of thing. So when cells use nutrients, it's a bunch of chemical reactions in series, and the term that we use that encompasses all of those chemical reactions that occur in a cell is metabolism. So you guys have used the word metabolism before, but that's the official definition of it, so you'll need to know that. So when we talk about these things, the primary source for all energy and all of life on Earth is our solar energy, like we talked about. And so that comes through in the producers, and those producers use that solar energy and go through photosynthesis. And photosynthesis basically just takes that solar energy and turns it into a chemical energy or organic nutrient molecules. So in this particular image, we're going to talk about two different things. So we have our nutrients, which if you want to think of nutrients more like food, and the chemicals that go with it, so the minerals that we need, the carbon-based sugars that we need, that's our nutrients. And if you'll notice here, those cycle through systems. So as I said, we have our primary producers, and then we have a bunch of different layers of consumers. So, our rabbits eat the plants, and then our hawks eat the rabbits, and so on and so forth, all the way up to us, who are tertiary consumers. And then either our waste, or when an organism dies, then the decomposers break down all that material and return it into the soil. It is then consumed by the plants, and we start all over again. So that's, a lot of times we'll talk about nutrient cycling or chemical cycling, and that's going to be true throughout any ecosystem. So our nutrients get recycled. The other part of this has to do with energy, and like I said, all life depends on solar energy. And so what happens with that is some of that energy gets passed from step to step, so our producers, our consumers, our decomposers, but part of it gets lost in the form of heat. And so because we lose that energy in the form of heat, we constantly need a new supply, and that comes from our sun. So that's why the sun is extremely important. And so your takeaways from this requirement is that nutrients are recycled, energy is lost at every stage, so constantly needs an input. You'll definitely want to know that all the chemical reactions within a cell is metabolism. Do at least understand that photosynthesis is the ability to capture solar energy and turn it into chemical energy and organic nutrients. So those are your key points for point 1. So our second requirement of living organisms is that they maintain homeostasis. And what homeostasis means basically is remaining in balance. So when you have an organism that is placed within an environment that's radically different from the inside of that organism, those inside features have to be maintained, and there are tight controls with feedback loops that allow for that. So biological systems need to maintain balance of humidity, temperature, other things like protein levels, for instance, salt levels, things like that. So this picture here really tells you how that's done. So if you'll look at this, we start with a stimulus that's produced because there's a change in a variable. Let's say, for instance, you've just gone on a run. Well, now you're thirsty. That thirsty is actually because you are dehydrated and there was a change in the available water throughout your system. So that's the stimulus that's produced. Then there's a receptor that detects that change. So you have sensors all throughout your body that are saying, do I have enough water or not? And when you've gone on that run and you don't anymore, then that information is then sent in our system to the brain. The brain is our control center, and that goes along what we call the afferent pathway. And that's a word that you're going to get used to, because if you do continue on in biology, particularly if you're going to go in the medical field, afferent and efferent will become regular words in your vocabulary. So information that says something is off balance or out of homeostasis travels along afferent pathways. For humans, that's along afferent nerves to the control center, which is the brain. And the control center is what says, this is what we need to do to fix it. And so then information is sent from the control center down efferent pathways, or efferent nerves for people, and they give an output that will then change something. And so that's the response to get things back into balance or back into homeostasis. And this occurs over and over again in organisms, even down to the smallest level, where you have individual bacteria. Clearly they don't have a brain, but they do have receptors, afferent pathways, efferent pathways, and an effector to make sure that homeostasis is maintained. Thirdly, living organisms respond to stimuli. So they have to be able to respond to their environment. And you'll notice that all of my examples here are plants. I am a plant person, so sorry about that, but it makes the most sense to me. So the top right is an image of, the common name is a sensitive plant. So they have these leaves, and once they get touched, they curl up like this. So it is responding to the stimuli of touch. Similarly, our Venus flytraps, once this fly lands in here, as you know, it shuts in order to be able to digest that particular fly. Now, both of those examples are what we call biotic factors, meaning that it's a living thing that has stimulated the organism and causes a response. There are also abiotic factors, and for that example, I have here a plant that's been turned on its side, and if you'll notice, the stems have grown in the upward direction. Now, they grow upward in response to light being up, but also in a negative response to gravity. Gravity is down, so even if I put this plant in complete dark, those stems would still grow up because they have the ability to grow in a negative direction to gravity. So both gravity and light, neither of those two are living things, so these would be abiotic factors. Our fourth characteristic of living things is that they reproduce and develop. So everything from egg stage to adult in our lovely grasshopper here, or even with human embryos, we go from sperm and egg all the way up to a living adult. Typically, we will call this the life cycle of a particular organism. So, in order for any particular species to survive, it needs to be able to reproduce or make another organism just like itself, or maybe not exactly like it, but a viable offspring is what we call it. So an offspring that's able to grow and develop and reproduce itself. So that's reproduction. Development is just that it proceeds through stages to change and become an adult. Obviously, we're going to take time later in this semester to really talk about reproduction and development, in particular looking at genetic material in DNA and RNA and how that's passed on from generation to generation, how it changes through mutations, and it allows for, those mutations allow for lots of diversity within a particular species, and so we'll get into that a little bit later in the semester. Lastly, we want to talk about that living organisms adapt. And so the official definition of adaptation is a change or process of change by which an organism or a species becomes better suited to its environment. So here we just have an example of a polar bear and how it is well designed for its environment. So it's large paws that keep it from sinking into the snow. If you think about wearing snowshoes, if you've ever walked in snow, if you're from the Southeast, that may not be something you're familiar with. I got to do that for the first time this year, so that was really cool. But it is a big deal to have this large surface area so that you don't sink into the snow. The white fur on the polar bears, yes, it keeps it camouflaged, but it also is for insulation. So each one of its fibers of hair is hollow, so it allows air to stay trapped in there and it holds the sun's heat. And so it's very well suited for its environment. Lots of small adaptations over time tend to change the look of a way a particular species is adapted to its environment. We can talk later about microevolution versus macroevolution. Things like this would be microevolution, so small adaptational changes that we can see. Another example of that is bacteria. It, through mutations, can sometimes be able to live in an antibiotic environment. That is a microevolution adaptation. So you see small changes in the population over time to better suit its environment. When people refer to macroevolution, that is where we start to see people thinking that those small changes all add up to huge changes and jumps into new species. But the important thing for us to know today is that living organisms do adapt. Now that you know the 5 characteristics of living things, let's test your knowledge a little bit. So on this page I have 6 different photos, some of which are living things and some of which are not. So let's start on the top left with the waterfall. I think many of you would have already said this is not a living thing, and that is correct. It obviously has energy, particularly potential energy from the water at the top being converted to kinetic energy as it gets released, but it can't reproduce, it's not made up of cells, so that is not a living thing. So moving on to the photo at the bottom left. Whew, that's ugly, right? This is an anglerfish. It lives in the deep ocean. It is a living thing. It is able to reproduce. It is able to maintain homeostasis, which is actually very tough at the bottom of the ocean. If you meet me in Bio 112, we'll talk a little bit more about that. But it is able to obtain energy. It actually uses this little thing at the top to act like a fisherman, acts like a lure and attracts other fish to it. So that is a living organism. Moving over here to our daisies in the center. These flowers, obviously they're not mobile, but they are able to meet all the 5 requirements for living organisms, so it is a living thing. If we move over here to the car, you'll notice that a car actually does meet some of the expectations, some of our requirements for living things. It responds to stimuli. You push the gas, it goes. Things like that. But we know that it is not a living thing because it can't reproduce. It's not made up of cells. Moving to the top right, this is bacteria. That is a single cellular organism. So it's small, but it meets all of our requirements. It kind of is the most basic of all those requirements, but it does meet the requirement of being able to reproduce, maintaining homeostasis. It's single cellular. It meets all of our requirements. Where things get tricky is this picture right here. This is a picture of a virus. Oftentimes people will think that a virus is a living thing, and it does have genetic material that it injects into its host, that the host then replicates the virus for it. But what makes it not a living thing is that, one, it's not made up of cells. It has a capsid, which is that outer envelope, has genetic material, has some proteins, but it is not a cell. So that alone says that it's not a living thing. It's also really not very good at maintaining homeostasis. So there's such a very quick turnover of viruses because they're very unstable. So a virus is not considered a living thing. So now that you are armed with the knowledge of what it takes to be a living thing, you know how to design experiments, we are going to move forward. So I'll see you next time.